Just to start with putting some faces to names with the 4 co-authors, and we're all on the line today. I'm going to use, skipping ahead there. So this project was a collaboration between the 2 networks of organizations noted here. The acronyms for our very long names are KIPCHE and OCDPA. Across these 2 networks, about 35 organizations were able to learn from each other and from the literature investigating 2 areas where our interests intersect: early life environmental exposures and the equally large topic of chronic disease. So a 2-year project stretched into 3 years when we realized the massive scope we had taken on, and we scaled back to a scoping review rather than a full literature review. The resulting report was published by the Canadian Environmental Law Association, the Environmental Health Institute of Canada, and the Ontario College of Family Physicians. Next slide. So I'll be using this outline slide to navigate across all 3 webinars. Today, introducing main findings and cross-cutting themes. The work was completed in June of 2011, and it's not been possible to stay on top of such a broad literature. Instead, we've aimed to keep track of the literature on endocrine disruption, and therein have focused on evidence about environmental links to obesity and diabetes, which will be the focus of the 2nd webinar. And nor did we address policy issues, though we initially thought we could. So like the 2nd webinar, the 3rd will bring us up to the present in terms of looking at policy implications. So I'm going to begin with a quick look at our overall scope and research methods. Next slide. We addressed multiple chronic diseases and potentially hundreds of environmental exposures. Both KIPCHE and OCDPA use the framing of the multiple determinants of health, but see important limitations in that framing that I'll talk about today. We looked at the developmental origins of health and disease concept, or DOHAD, as well as key challenges with how evidence is evaluated in environmental cases. That's a topic I'll get back to in the 3rd webinar. In looking at 5 areas of some of the most common chronic diseases, we focused on evidence about early life environmental risks, but we did so by first noting the broader context of what is known about multiple risk factors. And most important in terms of scope, we focused on existing comprehensive reviews, syntheses, meta-analyses published by credible and unbiased sources in the peer-reviewed literature, though we did also include direct review and reference to many individual studies. Next slide. So I'm going to begin with an overview of our main findings. First, there is indeed evidence linking early environmental exposures, most often prenatal exposures, with the later development of certain chronic diseases, though many knowledge gaps remain. For each of the chronic diseases we looked at, we also found limited to no recognition of environmental exposures in broader considerations of chronic disease risk factors. Going beyond the obvious and well-known risk factors, so diet, activity levels, smoking, alcohol, a look at multiple risk factors needs to consider over 3 decades of complex and interrelated changes, some of the context and trend information that I'm going to discuss in the next few slides. Within the continuum of shared, well-known risk factors across several chronic diseases, we note the primary importance of the social determinants of health, and as well this reality of shared risk factors points to the likelihood that environmental risk factors are also shared across multiple chronic diseases. And finally, a very wide range of chemicals or pollutants are implicated, with many of these arising frequently across several chronic diseases, and often those are substances that are already well-known problems. Next slide. So to begin then, a lot of context to carry through today and the 2nd and 3rd webinars. We have a high prevalence and or rising incidence of chronic diseases, several listed here, due partly to an aging population. Millions of people are and will be affected with staggering costs, and across the board the effects are worse for those living in poverty. Next slide. Adding more context, particularly when considering the important risk factors of exercise and diet, we are affected by major changes in the built environment and in our food production system with profound environmental impacts. We've not only changed the food production system, but food itself. Because of what we feed animals, as well as seeking extended shelf life for foods, we've changed the balance of fats in the animals we eat and in baked goods, and also massively increased our consumption of fructose. We have a loss of micronutrients in the soil and in the food. We're all familiar with this dietary shift noted in the third last bullet that has occurred in the context of increasing portion sizes as well as chemical contamination of foods from containers or packaging, pesticide residues, or general environmental contamination. So the latter would be particularly from methylmercury or persistent organic pollutants. And equality or equity issues arise again with all of these factors being felt or distributed more acutely among those living in poverty. Next slide. Across the same 3 to 4 decades, there's been a massive increase in exposure to synthetic chemicals and increased air and water pollution, although we've definitely seen some reductions there. Exposure pathways have also shifted. We've got a greater need to address exposures to chemicals incorporated into consumer products. And population-based biomonitoring studies confirm that we all carry low levels of many different chemicals, with the highest levels found in children. We often think of humans as being at the top of the food chain, but that place is actually occupied by our breastfed infants, who can be the most highly exposed. But I hasten to add that these findings should never deter breastfeeding, which is one of the many communications challenges with this information. And again, for many reasons, poverty increases these exposure risks. Next slide. Okay, 2 additional slides concerning child health and environmental factors. This slide is a summary of extensive evidence concerning the greater vulnerability of children. They are more highly exposed for reasons of proportionality and behavior, as noted in the first 2 bullets. This exposure can be magnified in the womb. We know this with methylmercury and bisphenol A, and it may be true of other substances as well. Fetal development is the time of greatest vulnerability, but postnatal and early childhood matters as well for development of the liver, kidneys, and immune system, and then development of the lungs and brain continues well into adolescence. And again, poverty increases the risk, creates greater risk for children, and First Nations children in Canada are likely at greatest risk. Next slide. So a final contextual slide is an overview of child health concerns associated with environmental exposures. As with chronic diseases, there are always multiple causes for any of these health outcomes. The knowledge base varies quite a lot across the impacts noted here. Just a general observation for the relatively small number of substances where we have strong evidence about associations with these outcomes in children, they are often the same substances where evidence is increasing about associations with later chronic disease. Next slide. So going back to the outline for today, I'll carry on with 2 cross-cutting themes. First is a 3-part task in this next section, the developmental origins of health and disease, or the DOHAD concept for short, underlying epigenetic mechanisms, and then key concepts related to endocrine disruption. Next slide. So very strong evidence tells us that the early environment, and I mean literally our first environment in the womb, affects lifelong health, directly influenced by maternal health, nutrition, levels of stress, etc. And this association has been observed literally since ancient times. Experimental work originated in 1989 with David Barker's findings in England and Wales of a relationship between low birth weight and subsequent early adult death from heart disease. Studies of the Dutch famine confirm this relationship, and other famines as well. During the so-called hunger winter of 1944, when food supplies to Holland were cut off by Germany, those who had been in utero had a high incidence as adults of insulin resistance, hypertension, metabolic syndrome, type 2 diabetes, and heart disease. And there's now very strong evidence of this association with epidemiological, clinical, and animal evidence. And the concept is expanding rapidly to address early life environmental exposures and their associations with these same chronic diseases, as well as several cancers. Next slide. The underlying cellular mechanisms are epigenetic, so meaning above or around the genetic mechanisms. All of our cells contain the same DNA, called our genome, but the wide variation in our cells and tissues is the expression of our epigenome. And so we're essentially talking about gene-environment interactions here. Change occurs during individual cell division or even across generations when genes are either turned on or off. The underlying DNA stays the same. These changes continue across our lives, which is why my sister and I, at 3 years old, looked a lot more alike than we do now. But this research tells us that cellular differentiation during the many complex stages of development is more strongly influenced by epigenetic than genetic mechanisms. So this diagram on the right is just the basic process of DNA replication during cell division to create proteins, and then that provides the building blocks for everything else during growth and development and maintaining homeostasis. So this is an oversimplification, but during any of the stages in this diagram, especially in the womb, there are epigenetic processes that increase or decrease the accessibility of DNA. So hence the definition noted here, where changes result from genes being activated or deactivated. And these mechanisms respond to the environment, what a child or a pregnant woman eats or drinks or breathes, even our behavior and experiences like stress during pregnancy, abuse during childhood, and poverty itself. A great deal of research demonstrates that the expression of the epigenome in brain development is affected by behavior and experience. Next slide. So we're talking here about chemical compounds attached to our DNA called epigenetic marks. And this illustration shows two ways that these chemical compounds, that is the epigenome, can mark or modify the genome and contribute to the regulation of DNA expression. So they play a role in turning a gene off or on. There's other ones too, but these are the two main ones that we know more about. So the first occurs when certain chemical tags called methyl groups attach to the backbone of a DNA molecule. And the second one is where a variety of chemical tags attach to the tails of histones, and those are spool-like proteins that package DNA neatly into chromosomes. So this action affects how tightly the DNA is wound around the histones, making the gene more or less available. So I'm noting this level of detail here so we're clear that scientists are increasingly able to understand the actions of chemicals, including toxic substances, at this most basic level. So recalling from the previous slide that the many stages of cellular differentiation that occur during development rely strongly on the epigenetic versus the genetic inheritance. So you can see why any disruption or interference with these mechanisms can have profound consequences. Next slide. So let's ground this back into the DOHAD concept. So I've mentioned there's very solid evidence showing that prenatal undernutrition alters gene expression through these epigenetic mechanisms. And the result is a loss of structural or functional capacity in the cells of key organs and muscles. This is permanent change. The manner in which these cells developed in an environment of malnutrition creates permanent vulnerability that in some cases can also be passed on to future generations if it's in the germ cell, but they're not genetic changes. So looking at latent diabetes risk, through epigenetic changes during prenatal development, these early environmental factors result in altered gene expression for how the pancreas develops and functions, and changes in metabolic processes related to glucose regulation and insulin secretion. These changes then contributed to that latent diabetes risk, often starting with a tendency towards midlife obesity and metabolic syndrome. Next slide. So finally, drilling down a bit further, we can see the influence of timing. Nutritional deprivation is the most profound if it occurs during the first trimester, less so in the second and third. Notice here that, however, that the latent tendency towards glucose intolerance occurs across all three, something to keep in mind in the context of other modern-day risk factors for obesity and evidence about obesogenic effects of endocrine disrupting substances, to be covered in more detail in the second webinar. Next slide. So I included the previous image because it was such a striking reminder of this one, an image that will be very familiar to those who know about the greater vulnerability of children to environmental contaminants. So this image is from a 1973 textbook about embryology, but it's been used for over 10 years to indicate prenatal vulnerability to toxic substances. As mentioned, in the womb, developing body systems and organs are highly susceptible to harm. During the first trimester, the embryo is susceptible to teratogens like alcohol and other solvents, methylmercury, and other toxic substances that can result in structural abnormalities or birth defects. But in the second and third trimester, when all that major morphological development is mostly completed, the fetus is susceptible to minor structural abnormalities and physiological defects, are more common. So things like impacts on brain function or later behavior. And again, that can result from exposure in the second and third trimester from, say, alcohol, solvents, or substances like lead, mercury, or PCBs. Next slide. So I'll move on now to a brief overview of endocrine disruption as another cross-cutting theme. Alongside 20 years of research into, more than 20 years, into the DOHAD concept, the science of endocrine disruption has also developed. And these two fields are now intersecting. It started with a multidisciplinary gathering of scientists in 1991 who initially coined the term endocrine disruption. Then the book Our Stolen Future and the website of the same name documented a wide range of observations in wildlife and in an increasing number of laboratory experiments. And by the time the Endocrine Society published this comprehensive review in 2009, they concluded that endocrine disruption can occur along diverse endocrine pathways, that these pathways are highly conserved in wildlife and humans, and that they can be modeled in vitro and in vivo. They also note strong evidence exists for adverse reproductive effects of endocrine disrupting compounds, and mounting evidence exists for effects in other endocrine systems, including thyroid, neuroendocrine, metabolic, as well as effects on insulin and glucose homeostasis. And finally, this recent issue of Endocrinology contains a further statement of principles focused on policy issues arising from this science. And I'll return to these latter two Endocrine Society statements in the second and third webinars. Next slide. Staying with an overview for now, some key observations. Endocrine disrupting effects occur at very low doses, and nonlinear dose-response curves are very common. Effects can be persistent and latent and result from early life exposure. Some examples are included in these images. Hypospadias is a birth defect with a suspected link to phthalate exposure, where the urethral opening occurs somewhere along the shaft of the penis and not at the tip. Increasing, though incomplete evidence, links prenatal exposure to bisphenol A in both breast and prostate cancers, the two most common cancers after skin cancer. And these mice, they were fed the same food. They had exactly the same activity levels. The only difference in this experiment was prenatal exposure to bisphenol A and resulting obesity at puberty or reproductive maturity. So in sum, we've got many different effects are possible, and many different chemicals are of concern. Next slide. And this slide does a good job of capturing two of these characteristics, so this ubiquitous exposure and the wide range of effects. You can see here that there's a wide range of substances, so categories range here across multiple kinds of pesticides, metals, and industrial chemicals. Not included are some pharmaceuticals. Biomonitoring data confirm that we are all exposed to most of these substances. And the color coding indicates just 4 of the endocrine pathways where endocrine disruption can occur. The Endocrine Society statement describes even more, and as does other evidence more recently in the literature. Next slide. So this image brings the DOHAD and endocrine disruption concepts together. In the second webinar, I'll describe some more examples. So for now, this simple image captures in the top level the normal developmental process where the genetic and epigenetic information in the cell creates the protein building blocks to create normal cells. And then the lower level, with the involvement of endocrine disrupting substances, gene expression is altered, proteins are altered or protein concentrations, and ultimately there is disease or dysfunction. So this could equally be an image of illustrating the DOHAD concept. So where it says endocrine disrupting chemicals, you could substitute inadequate fetal nutrition, excess maternal stress, and even the increasing evidence about similar epigenetic effects on brain development as a result of living in poverty. Next slide. To close out this overview then, I've noted here 3 useful journals that discuss early life environmental exposures within the DOHAD concept. The first 2 contain several really good literature reviews of key topic areas, and the third is a journal that started publishing in 2009 about the DOHAD concept more generally. This issue from 2011 is focused on the evidence about associations with environmental exposures. Next slide. So moving on then to an overview of the main focus of the scoping review. Looking broadly again, we situated the review of the literature in the framework of the multiple determinants of health. Next slide. and we did that for obvious reasons. We know that all health outcomes result from multiple factors, and chronic diseases are more complex and multifactorial than most. There's also a need to address the so-called big three, the behavioral or lifestyle risk factors, and their related biomedical factors such as hypertension, elevated blood sugars, high cholesterol, that sort of thing. These are very important risk factors, but they are not the full story. This framing it's a helpful and tidy circle, but it doesn't convey the primacy of the social determinants of health. This quote from Jeff Turnbull, who was then president of the Canadian Medical Association, summarizes a huge literature confirming that worldwide, health follows the gradient of socioeconomic status in rich and poor countries. The social determinants are often referred to as the causes of the causes. This reality of the primacy of the social determinants has been recognized in very strong statements from the World Health Organization, Canada's Chief Medical Officer of Health, and many others. And lately, even economists. The 2012 Global Risks Report from the World Economic Forum listed severe economic equality as among the top risks to future prosperity and security. Next slide. Okay, likewise for environment. That one little bubble labeled physical environment needs to capture a great deal, and environment is also a cross-cutting factor with many interactions, particularly with the social determinants. So as I mentioned, there are many examples of greater environmental exposure and health risks in low-income circumstances compared to the general population. There's two examples noted here. This fact is no different for climate change, the mother of all environmental issues now facing us. Another bubble in this frame, early child development, is also a huge and complex field, and it is also known to affect lifelong health, and it can be strongly influenced by poverty. Next slide. So moving on to how we reviewed the evidence, we looked at the conditions and diseases listed here, as well as a few areas I'm leaving aside today, including several types of cancer, asthma, and Parkinson's disease. We looked at known risk factors and then placed environmental risks in that context. An overall observation about known risk factors is the continuum that exists. From obesity through other aspects of metabolic syndrome, there are common and overlapping risk factors that can lead to type 2 diabetes, cardiovascular disease, vascular dementia, and Alzheimer's disease, which some researchers are referring to as type 3 or diabetes of the brain because of the involvement of altered insulin signaling and metabolism. And part of this overlapping picture is the fact that obesity and diabetes are themselves risk factors for other chronic diseases, including certain cancers, Alzheimer's disease, cognitive impairment, dementia, and cardiovascular disease. Next slide. So to narrow this down a little bit, let's just look at cardiovascular disease and type 2 diabetes. This division into non-modifiable and modifiable risk factors is typical in the literature. It comes from two very large international case control studies, InterHeart and InterStroke. There's so much overlap here with risk factors for heart disease, stroke, and type 2 diabetes that I've put them together for this overview. The so-called non-modifiable risk factors include gender, age, and the fact that some people have a genetic predisposition that can result in dyslipidemia, high blood pressure, et cetera. Those factors listed in the brackets, and basically the elements of metabolic syndrome. So-called modifiable risk factors, considered by these researchers to account for 90% of cardiovascular disease risk, so the InterHeart and InterStroke researchers. That includes the big three, so diet, smoking, inactivity, and again, those elements of metabolic syndrome that set up for either type 2 diabetes or cardiovascular disease or both. But note that it also includes stress. Next slide. The InterHeart team looked more closely at stress and saw a greater effect, making this conclusion highlighted in blue font that more stress and greater heart disease risk occurs when people perceive a lack of personal control. And these findings align with strong evidence about the primacy of the social determinants of health, where chronic disease risk factors are inseparable from living conditions and not necessarily modifiable when people are stuck in poverty. And then finally, the DOHAD concept is included in this overview of risk factors, and it calls into question as well whether genetic risk factors are strictly unmodifiable. From the DOHAD evidence, the idea of genetic predisposition may not be so deterministic when nutrition and stress and other life circumstances can have dramatic and intergenerational effects. All of this discussion of risk factors then provides necessary context in the scoping review for looking in the most detail at the evidence for risks posed by environmental exposures. Next slide. So finally, looking at environmental exposures. This slide summarizes a wide range of studies in adults looking at associations between various exposures and either risk factors for cardiovascular disease and type 2 diabetes or the diseases themselves. So in boldface are a range of substances. Each is followed by an indication in italic of the strength of evidence for the effects that are then noted. So for example, for air pollution, and this is mainly evidence about the criteria air contaminants, there is causal evidence linking air pollution and cardiovascular disease, though despite this strong evidence, it gets left out of the risk factors noted in the InterHeart and InterStroke studies. For low-level lead exposure, strong evidence exists indicating links to hypertension. And on down this list with a lot of animal data, some epidemiological data for the others that are noted here. And in blue font, you can see that effects are repeatedly about altered insulin signaling or related issues of inflammation and oxidative stress. And just to remind us here that insulin is one of the most important hormones in our bodies. It enables our cells to absorb glucose, transform it into energy. It's involved in blood vessel elasticity, so it has links to hypertension. It's involved in cognition and whole body homeostasis. So it's a very important hormone. Next slide. The epidemiological evidence in adults prompted a whole lot of animal research that indicates disruption of metabolic homeostasis through endocrine pathways, and greater risk or a more prominent effect is seen when exposure occurs in utero or perinatally. And these results, this is animal data, these results are a progression of the DOHAD research where, as I mentioned, there's a very solid understanding of the epigenetic mechanisms underlying latent hypertension, metabolic syndrome, and obesity as these arise when there is fetal undernutrition. I'll get into this evidence in more detail in the second webinar, but note here that where these associations are being seen between early environmental exposures and metabolic disruptions, the same kind of epigenetic mechanisms are beginning to be identified. Next slide. So looking at some of these exposures, this slide aggregates results of some key animal studies. 4 different endocrine effects to note here. First, the association between bisphenol A and permanent alteration of insulin metabolism. That elevates the risk for obesity, metabolic syndrome, and then the continuum of chronic diseases with these common risk factors. Second is a latent effect on adult testosterone levels, which may be a risk factor for cardiovascular disease and type 2 diabetes. The second one refers to evidence in animal studies for early exposure to bisphenol A and phthalates, and a human study showing this association between elevated PCBs in adult men and lower testosterone levels. A third one here, Bruce Bloomberg and colleagues have shown the epigenetic mechanism by which tributyltin influences the gene that regulates whether cells become fat or bone. This is research that was part of naming certain chemicals as obesogenic. Likewise, for the bisphenol A and phthalates research, epigenetic mechanisms are beginning to be described for these effects, and we'll get more into that in the second webinar. And finally, as mentioned on a previous slide, low-level lead exposure is known to be associated with hypertension in adults through neuroendocrine stress responses. In animal studies, there's also seen a synergistic effect of lead and maternal stress negatively affecting brain development, so for early life exposure to lead. So the effect of early life lead exposure on stress hormones may play a role in latent chronic disease risk, but because it affects the same neuroendocrine pathways, so that's the hypothalamic pituitary adrenal axis, HPA axis, and those pathways have been identified in the DOHAD evidence as playing a crucial role in that latent chronic disease risk. Next slide. So a reminder again, the next webinar is going to focus on some of this evidence in more detail. Given our huge scope, I focused on literature reviews and meta-analyses, as I mentioned before. I also looked at individual studies to dig further or to confirm details. I tried to focus on areas where a lot of evidence is accumulated, which is often the point at which meta-analyses can be done anyway. I had to leave out a lot of areas of emerging research, and as is so often the case in looking at environmental exposures, a lot more substances may be involved in these early life, often endocrine disrupting effects. So an overall point to make here is that we can look at two areas of very solid evidence. First, the DOHAD concept and its underlying epigenetic mechanisms, well-identified mechanisms. And second, the very clear understanding we have about links to chronic disease when metabolic processes are disrupted, particularly insulin metabolism. For early life environmental exposures, we have much less causal evidence, but an increasing body of evidence indicating that chemical exposures can operate through the same or similar epigenetic mechanisms to create the range of risk factors that we know are associated with hypertension, disrupted insulin metabolism, obesity, and other risk factors linked with cardiovascular disease, type 2 diabetes, Alzheimer's disease, and some cancers. Next slide. Okay, to wrap up this overview, and with apologies for the dense content of these next two slides, this comprises a single table across the two slides where we brought everything together. And the caveat in red font is very important. Strength of evidence for these associations is widely varied and not caught in this summary. I have only covered a portion of these results today, but across these two slides you can see in the first column the 5 areas of chronic disease we looked at. So cardiovascular disease, type 2 diabetes, and just to advance the slide, Alzheimer's disease, Parkinson's disease, 3 types of cancer, and a brief look at several other cancers where there is evidence of links to early life exposures, and finally asthma. So I'll just go back to the previous slide. The areas in purple are included for several reasons. Since cardiac birth defects are a known risk factor for latent cardiovascular disease, we looked at known or suspected causes from environmental exposures there. And low birth weights included because of the DOHAD evidence about fetal undernutrition. In fact, one literature review about metabolic syndrome suggested it be called small baby syndrome, given the strong evidence within the DOHAD concept of low birth weight setting up for latent chronic disease risk. We therefore included a look at evidence for environmental exposures that are associated with low birth weight. And we also separately noted chemicals suspected as obesogens for obvious reasons. Just advancing to the second half of this table, finally we did a very brief review of substances suspected in developmental neurotoxicity and found that many of the same substances suspected in developmental neurotoxicity arise elsewhere in this table. So leaving off many details here, it's notable that some chemicals or pollutants arise repeatedly across this table. These include air pollutions, mostly the criteria air contaminants, but also other toxic pollutants and environmental tobacco smoke. Metals, especially lead, bisphenol A, phthalates, several pesticides, solvents, and several persistent organic pollutants. So finally, I've really given short shrift in this presentation to the cancer evidence, and I'll do that again in the second webinar with the focus on suspected obesogens and diabetogens. The review and the report about the cancer evidence is one of the largest, and I can't do it justice in this overview. I'll just note here that cancer science in general is one of the areas where the understanding of epigenetics or gene-environment interactions is the most advanced. And I'll touch on these advances and their policy implications in the third webinar. Next slide. So a final recap here. We indeed have increasing evidence of these associations, although many uncertainties remain. The DOHAD, or developmental origins concept, is based on a very solid evidence base that is rapidly expanding to include early life exposures. Evidence for underlying epigenetic mechanisms can mean that prenatal or perinatal effects are permanent and therefore of serious concern. We need to put this information into that broad systemic context. Changes over the last 3 to 4 decades, much faster than changes in our genetic makeup can occur, have occurred in parallel with the rise in chronic disease, and they need to be considered, including multiple interactions. And finally, there is a continuum of shared and well-known risk factors across the conditions and diseases that I've looked at today. This reality combined with the increasing evidence for common epigenetic mechanisms in early life that underlie latent chronic disease risk points to the likelihood that early life environmental risk factors are also shared, including a role for environmental exposures and other early life risk factors like low birth weight. So as mentioned in discussing the large table in the previous 2 slides, Several chemicals or pollutants come up repeatedly, and they're listed here again, though a much larger list of substances is described in the report, and new research is published literally daily. So as you can imagine, I've only been able to touch the peaks of mountains of information here, so hopefully we can get into a bit more detailed questions in the next two webinars. Next slide. I'll end here and note that I've included a short list of some of the key references covered in the issues raised in these slides. And the slides are available on the CELA and KIPCHI websites, cela.ca or healthyenvironmentforkids.ca. And I will end there with some final thanks to colleagues and a few of the researchers whose work I've borrowed from more heavily than others, and pass it over to my colleagues for additional comments. Thank you very much.